Hi everyone, welcome back to Neuroscience for Non-Majors. This is week 7, unit 4, part 2, and I'll get right to it. I'll share my screen. Okay, so in today's class we will talk about memory consolidation, beginning with a breakdown of a paper that studied this phenomenon, that was done by Dr. Denny in Columbia University, I think. And we will also discuss sleep, some sleep disorders, and Alzheimer's disease. So just some quick review. As we know from our last class, the hippocampus is important for creating contextual and episodic memories. Within the hippocampus, the dentate gyrus takes very similar sensory inputs from the entorhinal cortex and rewrites them to be unique outputs that are no longer similar but still hold the same information. Then the categorized sensory information is sent to another part of the hippocampus called the cornu ammonis, or CA as an abbreviation, which is divided into 3 subfields. And through mossy fiber projections from the dentate gyrus, the sensory information goes to the 3rd subfield, CA3, where the dentate gyrus synaptic terminals have very strong synaptic connections. And also, as we learned in the last class, in CA3, a process called pattern completion happens, where the memory is processed in a way such that the person remembering it doesn't need all of the details to complete this pattern and understand what happened during the experience because of all the reciprocal interconnections in CA3. The memory is then able to be activated in the presence of a reminder. Again, as we learned in last class, during this entire process, a gene called ARC and another gene called cfos are expressed in the active areas of the hippocampus. ARC and cfos are immediate early genes, meaning they are expressed very quickly after receiving a stimulus, making their expression directly tied to neuronal firing. ARC and cfos expression are associated with synaptic plasticity, or how neural activity over time can change with synaptic strength. The strong synaptic connections between the dentate gyrus and cornu ammonis subfield 3 neurons are also crucial for synaptic plasticity. Another process that is very important for synaptic plasticity is adult hippocampal neurogenesis, which happens when neural stem cells in the adult dentate gyrus divide and turn into progenitor cells, which end up becoming neurons. As a common early symptom of Alzheimer's, lack of adult hippocampal neurogenesis and its effect are very important phenomena to study when trying to learn about age-related memory disorders. And also, adult hippocampal neurogenesis only happens in 2 parts of the brain, as far as we know, in the dentate gyrus of the hippocampus, which again needs a lot of cells in order to activate a unique group of neurons for every experience. And it also happens in the striatum. And neurogenesis is a process of newborn, so like newborn like neurons being born. And in infancy it happens, and then in adulthood, for the most part, you know, the neurons you have are the neurons that you're gonna have. Okay. So the Denny et al. Paper titled "Hippocampal Memory Traces are Differentially Modulated by Experience, Time, and Adult Neurogenesis" is basically trying to identify the locations of an engram in mice, determine how it responds to different contexts, evaluate its importance in behavioral expressions of a memory, and study how adult hippocampal neurogenesis or lack of adult hippocampal neurogenesis affects the expression of a memory. The results can be used to contribute to research into memory deterioration disorders and memory deterioration from aging. And so the experimenters began by creating a transgenic line where genetic material is inherited by offspring naturally of mice that had genes for both enhanced yellow fluorescent protein, EYFP, as well as a specific gene sequence which would cause cells that are immunoreactive for the immediate early gene ARC to express Cre recombinase, which we learned about in the previous class, and which would then be able to activate the yellow fluorescent protein after the mouse is injected with tamoxifen. By doing this, the experimenters were able to have mice whose ARC expressing neurons would be easily identifiable as they would be fluorescing. To create the line of mice, fertilized egg cells were injected with linearized DNA containing the gene sequence. Then the experimenters had to determine whether or not the yellow fluorescent protein would be a reliable marker for the memory expression related to the contextual fear conditioning that the mice were going to undergo. They set up a context A, where some mice would experience a foot shock and others would not. 5 days after the foot shock, mice were reintroduced to context A. The mice who experienced a foot shock in context A both froze more, which is their fear response, and they also had their dentate gyrus and cornu-ammonis subfield 3 neurons express the yellow fluorescent protein far more than the mice who did not experience the foot shock. This strongly suggested that the yellow fluorescent protein cells in the mice would be fluorescent with the contextual fear conditioning. Then, to determine how context affected the engrams in the hippocampus, a group of mice were all given foot shocks in context A. Then, 5 days later, some mice were reintroduced to context A while others were introduced to a new context B. The mice showed far more freezing in context A than in context B, and the number of co-labeled immediate early gene yellow fluorescent protein cells that fired was much greater in the mice that were put back into context A. During encoding of the memory, the number of yellow fluorescent protein cells was greater in the dentate gyrus than in cornu-ammonis subfield 3, suggesting that the dentate gyrus is more important for encoding the memories. The effects of remote versus recent exposure to the context was also tested. After 30 days, mice froze in similar ways in both contexts A and B. In the dentate gyrus, the number of co-labeled immediate early gene and fluorescent protein cells did not change between the 5-day wait and the 30-day wait, but the cornu-ammonis subfield 3 had a decreased number of these fluorescing cells in the 30-day wait group compared to the 5-day wait group. This shows how the memories were moved out of the hippocampus as time went on and were no longer localized in CA3 anymore after 30 days. And they're also correlated with a lessened ability to discriminate between the real context where they experienced the fear and the similar context where they did not experience the fear. This generalization process occurs with the transition from short-term memories to long-term memories. It might seem like a negative because, at first it might seem like a negative because we remember fewer details with time, but actually it's really important to make sure this generalization happens so that we can identify similar contexts and come to a reasonable conclusion that if we experienced fear or some emotion or thing in one context, there's a likelihood that we will also experience it in the similar context. So next, the mice were bred to also have genes that would give their neurons light-gated inhibitory ion channels, so that through optogenetics, which we learned about last class, the experimenters could hyperpolarize the dentate gyrus neurons and CA3 neurons, preventing them from firing by using fiber optic cables that go into the dentate gyrus or CA3 and can direct light into the correct areas and trigger the light-gated ion channels to bump down the charge of the neuron. The goal of this was to see what inhibiting the labeled cells in the hippocampus would do for the behavioral expression of the fear memory. So 2 weeks after being given the contextual fear conditioning, mice were put back into the fear-inducing context A, but with the fiber optic cables that could inhibit the neurons that were tagged during the fear memory consolidation inserted into their hippocampuses. So the first 3 minutes of reintroduction to the context A had the light on. So with the light on, there's inhibition of the fear memory cells. And compared to minute 4, which is light off, and during the light off, there's no inhibition of the fear memory cells. So so the first 3 minutes had light on, the following 3 minutes had the light off, So there was a much lower amount of freezing in context A with the optogenetic inhibition compared to without the optogenetic inhibition, comparing minute 1 to minute 4. So after the optogenetic inhibition stopped, the freezing levels were similar to those shown in the mice who had not had their neurons optogenetically inhibited. The mice were also introduced to novel context B with optogenetic inhibition, but even with the optogenetic inhibition, there was not a significant change in the mice behavior with or without inhibition in the different context B. So this shows that the engrams in the hippocampus are crucial for behavioral expression of a fear memory in mice. The experimenters also tested partial suppression of the CA3, but found that unlike the result when all the marked cells in CA3 were being inhibited, when the CA3 is only partially inhibited, memories are not prevented from being expressed. So the final part of the experiments dealt with adult hippocampal neurogenesis and its effect on memory expression and consolidation. So using X-irradiation, adult-born hippocampal neurons in the mice were removed, and the X-irradiated mice ended up freezing a lot less in fear context A, suggesting impairment in memory expression. In terms of fluorescence, while in the dentate gyrus, the reactivation did not change, in the CA3, there was a decreased reactivation in context A for the X-irradiated mice. Social defeat was another strategy where you put basically a small mouse with a big mouse that intimidates it. And it was used to create depressive symptoms in the mice. And depression also decreases adult hippocampal neurogenesis. So the mice exposed to social defeat after 10 days were given the contextual fear conditioning, and where again, they're given a foot shock and they're trained to associate the foot shock with the context. And they basically had decreased reactivation in CA3, but not dentate gyrus, just like the irradiated mice. So this suggests that with the exception of a remote memory reactivation, so like that's when the memory is not in the hippocampus anymore and has been transferred to the neocortical storage sites. So with the exception of that, their level of reactivation in C3 is correlated with how powerful the engram is. So the neurogenesis results are interesting because even though adult hippocampal neurogenesis only happens in the dentate gyrus, the percentage of reactivated cells in C3 is reduced instead of the percentage of reactivated cells in dentate gyrus. All of these experiments allowed for identification of and demonstration of the importance of engrams in the dentate gyrus and core neuromonas subfield 3 for behavioral expressions of memory in different contexts, as well as highlighting the ramifications of manipulating the neurons associated with pattern completion and pattern separation in various ways, particularly to optogenetically inhibit the crucial memory traces to learn the extent of their importance, and to study a mouse model of lack of adult hippocampal neurogenesis. All right, so remember how in the study, the memories were no longer localized in CA3 of the hippocampus after time had passed, and they had been transferred to neocortical storage sites. And along with this transfer came a generalization of memories where they couldn't distinguish between context A and context B as well anymore. So what exactly is the cause of this? Well, it turns out that during sleep, memory consolidation takes place. This selection, transfer, and strengthening of memories as they go from short-term to long-term storage sites. We can categorize sleep by REM or REM, which stands for rapid eye movement, and NREM or non-rapid eye movement stages. And they cycle with one another. You can kind of think of being awake as the reception state, where you're receiving all sorts of inputs and experiences. Then you can think of NREM as the reflection state, where unnecessary neural connections get pruned away. And then REM finally is the integration stage, where the connections that survive the pruning processes of NREM are strengthened and integrated with other brain activity patterns. So NREM is characterized by very slow, coordinated brain activity, slow wave sleep, that allows distant parts of the brain to communicate effectively, again, because of the synchronization of neural activity across the brain, causing this particular waveform called a delta wave. A brain structure called the thalamus You can think of it as the sensory gate of the central nervous system, which is able to prevent or enhance signals sent throughout cortex, is what drives these slow waves. During sleep, the thalamic neurons, which project to cortex, suddenly transition from being constantly active to firing in like bursts. And these bursts of activity cause downstream bursts of activity as well, these slow synchronous waves allowing distant brain areas to communicate. And during this synchronous activity, memories are actually becoming less dependent on the hippocampal indexing circuit, and after months of this process, are able to reactivate in the neocortex without needing the hippocampus as much. During REM sleep, short-term and long-term memories are being replayed and integrated, and this replaying of memories is what dreams are. This is also why REM brainwaves look like awake brainwaves, very quick and with much less synchrony compared to NREM brainwaves. They are, after all, replaying experiences. During REM sleep, your eyes move around very, very quickly, hence the name rapid eye movement sleep. So again, during REM, you don't want to be acting out your dreams, right? There needs to be something that disconnects your motor activity from your dreams. This disconnection is caused by a neurotransmitter called serotonin, which binds to a receptor in a brain structure called the pons and prevents motor signals from reaching the spinal cord. The pons affects all motor signals except for your eye motor signals, so your eyes are free to move around during REM. Stroke damage to the pons causes something called locked-in syndrome, where you're unable to move any part of your body except for your eyes. Similarly, sleep paralysis occurs when you wake up, but that serotonin projection to the pons has not shut off yet. So you're unable to move despite being awake. Sleepwalking, though, actually occurs not during REM, but during the deepest stage of NREM, and its causes are still being debated. Also, insomnia is another important sleep disorder, and it's actually caused by abnormal circulation of cortisol, which is a hormone associated with stress. So in the morning, we generally have a sharp cortisol peak that helps us wake up, but if cortisol levels are high when we're trying to sleep, then we'll be unable to fall asleep or be unable to have a restful sleep. In depression and anxiety disorders, which we'll talk about in future classes, those disorders are characterized by higher cortisol levels, and insomnia is often a symptom for both of those. We also need to discuss melatonin and adenosine. So a part of the brain near the optic nerve intersections, called the suprachiasmatic nucleus, receives signals from cells in your retinas. When photons hit the retina during the daytime, which is full of light, these retinal cells excite the suprachiasmatic nucleus. When the retinal cells excite the suprachiasmatic nucleus, through intermediary neurons, inhibits a gland called the pineal gland and prevents the pineal gland from releasing melatonin, which is a chemical that promotes sleep. When it is dark, however, the suprachiasmatic nucleus is unable to inhibit the melatonin production because the retinal cells are not receiving light. So during the dark, melatonin circulates a lot more and is able to promote sleep. Another chemical, adenosine, is also super important for this sleep pressure feeling. Adenosine levels slowly start building up during the daytime, and then when you sleep, the levels fall away. Caffeine works by binding to metabotropic adenosine receptors instead of the adenosine molecules. So the adenosine is not able to activate the receptors. But the adenosine is still building up nearby, so as soon as the caffeine wears off, a whole bunch of adenosine binds and causes you to experience a caffeine crash. Another thing that happens during slow wave sleep is the increased circulation of cerebrospinal fluid in the brain. The extracellular space between neurons increases during sleep and causes less resistance and increased flow of cerebrospinal fluid through this new extra space. The cerebrospinal fluid, or CSF, picks up waste products and junk proteins and takes them across the blood-brain barrier, where they then travel through the bloodstream to the spleen and to the liver to be cleaned out. One of these proteins that is cleared out through this process during sleep is called beta amyloid, which can aggregate dangerously and form plaques that cause cell death in Alzheimer's disease. There are two causes of Alzheimer's, these amyloid plaques and tau tangles. So tau tangles are caused by misfolding of microtubule proteins inside a neuron. Microtubules normally help with nutrient delivery to the cell. Tau proteins normally help regulate microtubule function. But in Alzheimer's, this regulatory tau protein gains a lot of extra phosphate groups, and it suddenly creates tangled up misfolding microtubules. When they become misfolded, the tau tangles become this thing called a prion, which means they now have the ability to cause other microtubules to misfold too. This creates an ever-growing chain of misfolding microtubules that begins to travel from one part of the brain to another, obviously affecting the ability of the cell to receive crucial nutrients. The buildup of that beta amyloid protein also causes increased phosphorylation of the tau proteins, so there is an amplification effect as the amyloid plaques and tau tangles both build. In Alzheimer's disease, the first cells to be affected are the medial temporal lobe cells, like including hippocampal cells. And this causes huge memory deficits in ability to form new memories as well as remembering older memories. As we age, we have poorer sleep quality and shorter length of sleep and a shifted circadian rhythm. And all of these factors do affect the ability of the cerebrospinal fluid to flush out the amyloid plaques, which is why Alzheimer's occurs often in old age. All right, and that's it for this class. The mid-semester projects are going to be due next week, and we'll have like a gallery walk sort of thing over Zoom where we can all like see each other's projects. And these were the references for this class. All right, see you soon.